The cell cycle length depends on the type of cell. In embryonic cells, it's rapid. In epithelial cells, which cover organs and surfaces in mammals, they can replicate every couple of days. Still others remain in the G0 stage indefinitely, like muscles and neurons. The majority of the cell cycle takes place in interphase. The cell cycle is controlled by either external or internal factors. Externally, cells are either inhibited or stimulated to grow based on nearby damaged tissue, release or inhibition of human growth hormones, crowding of cells, and other things that can inhibit cell division. Regardless of the external conditions, cells must pass several internal checkpoints before they can divide into two. Mistakes in DNA are called mutations. The mother cell has several mechanisms to reduce the risk of mutations as it divides into two daughter cells. There are three distinct cell cycle checkpoints during the 4 phases of the cell cycle. Interactions among regulatory molecules at each checkpoint allow a cell to decide whether to proceed with division. If these regulatory molecules are defective, the checkpoint may fail and cells may start dividing in an uncontrolled fashion. The G1 checkpoint is the most important. This checkpoint determines whether the cell will continue through the cell cycle and divide or exit the cycle and enter the G0 stage. 4 factors affect whether the cells pass the G1 checkpoint. 1 is cell size. Cells must be large enough to split into 2 functional daughter cells. 2, there has to be plentiful nutrients available, as this is an energetically costly process. Next, DNA has to be undamaged. To determine this, there's a special protein called the p53 protein, which either pauses the cell cycle or initiates apoptosis, which is programmed cell death, if the DNA is physically damaged. p53 is an example of a tumor suppressor protein. Damage to the p53 gene can actually lead to uncontrolled cell division, but we'll talk more about that one later. Finally, there's social signals from other cells. So cells in multicellular organisms pass or do not pass through the G1 checkpoint in response to the signaling molecules from other cells. For example, if you're injured and you got a cut on your skin, the social signals will stimulate cell cycle growth, whereas if the injury closes up, then you're going to see social signals that stop the process of cell division. The second checkpoint is the G2 checkpoint. This one takes place during the G2 and the mitotic phases. Cells stop growing here if the chromosome replication has not proceeded properly or if the DNA is damaged. Finally, the third checkpoint is the metaphase checkpoint. Cell growth ceases during metaphase if the chromosomes are not properly attached to the mitotic spindle. This mechanism prevents incorrect chromosome separation that could give daughter cells the wrong number of chromosomes. The 3 cycle checkpoints prevent the division of cells that are damaged or that have other problems, and they prevent the growth of mature cells that should stay in the G0 state. In addition to the checkpoints, there are regulatory molecules that initiate and inhibit cellular division. These molecules can either promote cellular division, these are called positive regulation, or inhibit cell division, called negative regulation. Cyclins and cyclin-dependent kinases are 2 positive regulators for cell division. When combined, this is called the cyclin-dependent kinase, or CDK, complex. These are inactive while 2 phosphates are attached to the enzyme. In order to be activated, a phosphorylation event must occur in which an ADP picks up one of the phosphates, and this will allow the continuation of the cell cycle. There are several cyclin proteins, but there is a strong correlation between high concentrations of cyclin and passing checkpoints, once cyclin levels decline and pick back up as you approach a new checkpoint. There are times during the cell cycle where it must be halted due to environmental or internal conditions. In that case, a CDK inhibitor blocks cyclin, preventing its progress from attaching, and will remain on CDK until the issue is resolved. Negative regulators inhibit the cell cycle. So far, biologists have discovered 3, RB, P53, and P21 proteins. All of these classes of proteins are called tumor suppressor proteins. P53 and P21 proteins are used during the G1 checkpoint to ensure the DNA is undamaged before it copies itself. If the DNA is damaged, it will then signal the proteins to fix the DNA before continuing. Typically, a DNA polymerase has to proofread the DNA for any errors before continuing. If the DNA is irreparable, P53 triggers apoptosis, which is programmed cell death, whereby the cell will destroy itself to prevent the spread of faulty DNA. It's important to note that apoptosis is also used to destroy cells infected with a virus. Cells respond to signals from other cells so that they can divide, and only when their growth benefits the whole organism. This is known as social control. Social control is based on growth factors, which are polypeptides or small proteins released by cells that stimulate division in other cells. Generally, cell cultures will not grow unless growth factors are present. So think of these as on-off switches. Cancer cells, however, divide without growth factors. They are no longer subject to social control at the G1 checkpoint. RB proteins, also known as retinoblastoma proteins, enforce the G1 checkpoint, keeping the cell in the G0 state. A transcription factor called E2F is removed from DNA, preventing the cell from leaving the G0 state. When E2F is returned, the cell cycle can continue. And the only way to do that is to phosphorylate the RB protein, and by doing so, it releases the E2F transcription factor, allowing for the cell cycle to continue. Excessive growth factors can override the inhibitory effects of RB. Cyclin synthesis is triggered and cyclin CDK complexes are activated, leading to the activation of any of those S-phase proteins that help with the replication of DNA. In some human cancers, the G1 cyclin is overproduced, permanently activating CDK, which then continuously phosphorylates its target proteins, meaning more division. Either excessive growth factors or cyclin production in the absence of growth factors can cause cyclin overproduction. Which brings me to my next point, cancer. Cancer is a class of diseases that all relate to some failure in the cell cycle, such as passing checkpoints with faulty DNA, malfunctioning tumor suppressor genes, or just the mutation of proto-oncogenes into oncogenes. Most cancers are gradual, as it starts with minor errors in DNA that accumulate as the cells divide. Eventually, the cell cycle speeds up as the control of the cell cycle and repair of undamaged DNA or proteins no longer works. This uncontrolled growth of mutated cells can lead to tumors, and left unchecked, they will eventually metastasize or spread to other parts of the body. And just a point of note, not all mutations are bad. In fact, many of the adaptations that we have, like opposable thumbs, all started off as a mutation. And so when a mutation is positive and majority of the population ends up inheriting it over time, we call that an adaptation. But ultimately, cancer mutations are always bad. Genes that encode for positive cell cycle regulators, such as cyclins and cyclin-dependent kinases, are called proto-oncogenes. Everyone has these, and are normal genes found within our genome. However, they can become mutated into oncogenes, which then lead to cancer. Most of the time, when oncogenes form, the cell cycle shuts down for that cell, but some mutations can cause oncogenes to overproduce cyclins and cyclin-dependent kinases to speed up the cell cycle. Most often, this comes from these positive regulators speeding up the process to the point that the cell passes checkpoints with mutated DNA. And sometimes they result in cells that can't reproduce due to these changes, but if a daughter cell can successfully be made and reproduce, it can lead to cancer. Tumor suppressor genes are portions of the DNA that encode for negative regulator proteins such as P53, P21, and RB. These prevent the cell from furthering the cell cycle until checkpoints are met. Cancer can arise when a tumor suppressor gene is mutated, causing it to no longer set up the roadblocks, forcing the cells to pass through checkpoints, therefore faulty cells continue to divide. The ultimate nightmare scenario is when proto-oncogenes mutate to oncogenes and tumor suppressor genes lose functionality. Now the cell has no control over the cell cycle and cancer ensues. Cancer starts with mutations in the proto-oncogenes and tumor suppressor genes. Many cancers are brought on by genetics, so if your parents or grandparents have cancer, your chances of having cancer increase. There's several environmental causes of cancer. We call these carcinogens. These include smoking, asbestos, poor air quality, certain chemical exposure, ultraviolet exposure, etc. All of these cause mutations in the proto-oncogenes and the tumor suppressor genes, which bring about cancer. Cancer starts off with a cell that is mutated to the point that none of the regulatory molecules are functioning. If that cell survives cellular division and is missed by the immune system, because one other thing I wanted to mention, the immune system can attack cancer cells. But if this one's missed by the immune system, it will then be rapidly divided to form a tumor. Tumors are living masses of your own cells, and they require nutrients and gas exchange, and so growth factors are sent to the blood vessels to help feed this tumor. This is called angiogenesis. As cancer progresses, the cancer cells can then enter the bloodstream and spread, or metastasize, throughout the body. This is when cancer becomes difficult to control. The success of cancer treatments depend on the following. How far along is this cancer? If you spot it early, you can prevent metastasis or go into remission, meaning cancer is under control. Curing cancer is impossible because it is a self-genetic disease. You can only suppress it throughout your life. How close the cancer is to the surface of the body is also helpful. Skin cancer is much easier to treat than pancreatic cancer because skin cancer is readily visible, whereas pancreatic cancer requires MRIs, X-rays, and other forms of technology. Age. Unfortunately, as you age, your cells are more likely to produce faulty DNA. So the older you get, the harder cancer is to control. The more your cells divide throughout your life, the higher the likelihood your cells will produce defective daughter cells. Also, the state of your immune system. Treatments tend to weaken all of your cells, including the immune system cells, so it makes you more prone to contracting other illnesses. Finally, how do prokaryotic cells divide? So bacteria do not undergo mitosis due to having no nucleus. The nucleoid, which is shown in the red circle, copies itself and distributes itself to the opposite sides of the bacterium. It then develops a septum to divide the cells, allowing the cells to break off of one another. So that septum is a portion of the cell wall. Some single-celled eukaryotic organisms also undergo binary fission as a means to asexually reproduce. While this and cellular mitosis can lead to making more cells, all the cells are identical to each other. However, the key to life is genetic diversity. More diversity of genes amongst the population increases the chances of a species to survive changing environmental conditions. But how do living things achieve that? Well, that's the topic of our next chapter, meiosis and sexual reproduction.